Welcome to AI Explains It All, where we break down complex science into bite-sized knowledge. Today, we're diving into the amazing process of DNA replication. DNA replication is how cells copy their genetic information before dividing. It all starts with the double helix, the famous twisted ladder structure of DNA. The two strands of this helix are made up of nucleotide pairs, A with T and C with G. But how does this complex molecule replicate itself? First, an enzyme called helicase unwinds and separates the two strands of DNA, creating a Y-shaped area called a replication fork. Each strand of DNA now serves as a template for a new strand. That's where another enzyme, DNA polymerase, comes in. It attaches to each original strand and starts adding new nucleotides, building a complementary strand that matches the original, A pairs with T and C pairs with G. One strand, called the leading strand, is built continuously. The other, the lagging strand, is built in small sections called Okazaki fragments, which are later stitched together by an enzyme called helicase. This ensures that both strands are replicated accurately. Once the process is complete, two identical DNA molecules are formed, each with one old strand and one new strand. This semi-conservative process ensures that genetic information is preserved during cell division. And that's how DNA replicates itself, enabling life to grow, heal, and evolve. Thanks for watching. If you found this explanation helpful, don't forget to like and subscribe for more science breakdowns. Got a topic you want us to explain? Let us know in the comments.